Cells are responsible for all of our functions, and as such, they need to transport materials in and out of the cell all the time across the cell membrane. For instance, oxygen has to enter the cells for cellular respiration, and the waste product of that process, CO2, needs to be able to pass out of the cell across the cell membrane constantly as well. In addition, sugar, glucose, is needed for energy. Ions are needed to enter the cell. Ions need to be pushed out of the cell, some waste ions. So as you can see, there are a lot of things that have to move across that cell membrane. Now, that cell membrane is referred to as a semi-permeable membrane with regards to cell transport. Small and uncharged molecules like oxygen gas, O2, as you can see here, are able to slip easily through that phospholipid bilayer. CO2 as well, coming out of the mitochondria there, slipping easily across the cell membrane because it is small and it's not polar or charged. Now, large molecules like starch have a hard time crossing the cell membrane. In fact, they can't because they are just too big and they will bounce off of the plasma membrane, so to speak. Other molecules or individual elements that can't pass through a semi-permeable membrane are those that are charged, like you see here, the sodium and hydrogen cations. Because of their charge, they are unable to pass easily through the phospholipid bilayer. Because again, the phospholipid bilayer is non-polar, so anything that's charged won't be able to pass through. So the rule here is large or charged molecules cannot pass through easily or simply. They must be let through through special proteins or through the use of energy along with those proteins. Also, these charged items that can't get through aren't just charged in the ionic sense, but also anything that's polar is unable to get through that phospholipid bilayer. Okay, so to start understanding some of the forms of cell transport, we need to become familiar with some of the vocabulary associated with concentrations. In particular, we need to know what a solution is. It is the combination of a solute dissolved in a solvent. So if we're to take a simple glass of water, the water would be the solvent, and in biology that is most often the case. Most solutions are made of water. Now, if we were to sprinkle some sodium chloride in there, our table salt, remember that this is an ionic bond and that water is an excellent solvent that will tear that ionic bond apart, leading to the individual ions. Then we would refer to these sodium and chloride ions as the solutes because they are what is dissolved in the solvent. Together we have a solution, a salt solution. Now, solute concentration is something we're going to talk about a lot as we move forward in talking about cell transport. And that is simply the quantity of a solute in a given volume of solution, usually expressed as mass over volume, milligrams per liter, or something like parts per billion. And the area of higher and lower concentration will be very important. There are two main types of transport. Passive transport is the type where no energy is used to move materials back and forth, and active transport is when the cell does need to exert some energy. So let's consider the simple passive transport situation first. And the first type of passive transport we'll explore is simple diffusion. So let's say we have a drop of red dye that we're going to drop into this glass of water. When we do that, it will disperse into its individual dye molecules. Now, simple diffusion is the movement of a solute from high to low concentration. So the red dye here is our solute, and you can see that at this moment in time, that little cluster of red dye molecules is an area of high concentration, high solute concentration. So they will disperse throughout the entire beaker of water, moving from high to low concentration. That movement, we say, is traveling down the concentration gradient. So all those other areas of the glass that didn't have the dye molecules were areas of low concentration, and the dye molecule went there. Now notice that the molecules do disperse evenly, and they will the net diffusion will stop at that point where there's equal solute concentration throughout the solution, and this is what we call equilibrium. In our cells, for instance, because they need oxygen, we can see the same thing happening here, where oxygen is passing by outside our cells in our bloodstream, and that would be an area of high oxygen concentration. Inside our cells would be an area of low oxygen concentration. So the O2 molecules diffuse down that concentration gradient until we reach equilibrium. Now in our cells, they will use that O2 up, and therefore maintain the concentration gradient, high outside, low inside, and thus O2 molecules will continue to diffuse in, get used, and more O2 molecules are always streaming by to be taken in.